We will discuss about genetic code now. What is genetic code? It is a dictionary that corresponds with the sequence of nucleotides and sequences of amino acids. That means nucleotides are present in the sequence and these nucleotides will code for amino acids. That code is called the genetic code. Code means they will give a code about which amino acid has to be made, 3 nucleotides. So that is called a genetic code. Genetic code is a set of rules by which information encoded in the genetic material, DNA or RNA sequences, is translated into proteins by the living cells. So it is that in the genetic code, the DNA or the RNA sequence is translated, or the genetic material is translated in the form of proteins. So this term genetic code was given by George Gamow. To understand how proteins are encoded began after the structure of DNA was discovered by James Watson and Francis Crick. George Gamow postulated that the 3-letter code must be employed to encode the 20 standard amino acids used by the living cells to build proteins. He said that to code for the 20 amino acids, we need a 3-letter code. Why is it that? I will explain here. The letters A, G, T and C correspond to the nucleotides found in DNA. So these are the nucleotides A, G, T and C. They are organized into codons. Obviously these only will be organized. The collection of codons is called genetic code, and this codon collection is called genetic code. For 20 amino acids there should be 20 codons. Yes. Each codon should have 3 nucleotides to impart specificity to each of the amino acid for a specific codon. Why is it? Because one nucleotide will give 4 combinations. That means only 4 amino acids will be coded. 2 nucleotides will give 16 combinations. That means only 16 amino acids will be coded. When we take 3 nucleotides and 64 combinations will be there, and it is more suited for 20 amino acids. That means all the 20 amino acids are coded. Now, the genetic code which is transferred from the DNA into the RNA in that 3 base pairs, like here ATG codes for 1 amino acid. Okay. Similarly CTA codes for another 1 amino acid. GGC code for another amino acid. So these are all present on this ladder of the RNA. But there is no comma, there is no, these are characteristics we will read later. Now genetic code. This is a table of all the genetic codes, all the 64 amino acids which are produced from the genetic code. See, we have to make 3 codons. That means we have to make 3 codes which will make 1 codon. Okay. So 3 codes are the first position of the code U, we have taken U, C, A and G. Similarly here also the second position of the code U, C, A and G. And the third position of the code will be in one block only, in the U block UCAG, similarly in C block UCAG, and in A block again UCAG, and G block UCAG. Now this U is here, U. The second U is in the second position. The third U from this side, here, this is the third U. This U is here, and we got one code for phenylene. Then U first one, the U second one, and the C of the third position has come here, and we get UUC. Okay, uracil and cytosine. Then U first position, U of second position, and then A of the third position. From the third position we will take A. We will get UUA, the third code. Similarly U of first position, U of the second position, and G of the third position here. It will give us the code UUG. So it codes for the amino acid leucine. Now here in this also you can see that 2 codes are there, UUU and UUC, which code for phenylene. Similarly, UUA and UUG code for leucine. So there are 64 codons. Okay, here you can count, there are 64. Out of these 64 codons, this is the table, how it is made. It's a very simple table, you can also make it. Then the genetic code and its types. It is a dictionary consisting of genetic words called codons. Each codon consists of 3 bases. There are 64 codons. And now out of the 64 codons, 61 are for 20 amino acids and 3 codons do not code for any amino acids. Here you can see stop codon, stop codon, stop codon. These are the 3 codons which do not code for any amino acid. So when they come on that mRNA, then the protein making of protein stops there. So they are also called stop codon or nonsense codons. Then talking about the types of codon: sense codons, signal codons, start codon, stop codon, and terminating codon and initiation codon. So we will talk about these 2 types, sense codon and signal codon. The codon that codes for amino acid is called sense codon. Those codon that code for a signal during protein synthesis are called signal codon. Now these signal codons are 2 types, terminating and initiating. That means those give the signal that protein synthesis must start now, like the terminating codon and the initiating codon. Terminating codon, as I told you, the stop codons are UAA, UAG, and UGA are terminating codons or nonsense codons and are referred to as amber, ochre and opal. The initiating codon is only one and it is for the first amino acid in all proteins. It is the same. It is AUG. It codes for methionine both in eukaryotes and in prokaryotes also. Then coming to anticodon. Anticodon is a sequence of tRNA. On the tRNA you can see anticodon is there, and which pairs with the codon of mRNA, okay, during the translation of protein. So difference between codon and anticodon. Now, what is a codon? What is an anticodon? Codon is present in both DNA and RNA, but anticodon is always present in RNA. Codons are written in 5 to 3 direction. Anticodon is written in 3 to 5 direction. Anticodon of some tRNA molecules have to pair with more than one codon, because more than one codon codes for different for the same amino acid. Then codons are sequentially arranged in nucleic acid strand, while anticodons are discretely present in the cells with amino acid attached to it. So codons are present on the mRNA, while anticodons are present inside the cells, and they have to be collected by the tRNA and then bring them together and then form a protein, help in making of protein. The codon defines which anticodon should come next. So because mRNA, the codons present on the mRNA are in a sequence. So when 3 codes will code for one amino acid, the next 3 codes determine now which anticodon will come and join them, okay, for the next amino acid which is going to be created. Anticodon helps in bringing a particular amino acid at its proper position during translation. So the anticodon will bring the amino acid in the particular position. That anticodon is there at the lower part of the tRNA, and at the upper end of the tRNA, at the 3' end, there is the amino acid attached. Okay, so that amino acid gets attached into a chain that we will read later. Then coming to characteristics of genetic code. These are the various characteristics: triplet code, comma-less, non-overlapping, coding dictionary, degenerate code, universality, non-ambiguous, chain initiation code, chain termination codons. One by one we will read about it. What is triplet? The genetic code is a triplet. There are 64 codons. So you can see it is always a triplet. Always there should be 3 nucleotides attached. Then universality. The genetic code is universal. That means it is same for eukaryotes, for prokaryotes, for everyone. AUG is a codon for methionine in mitochondria also. It is in cytoplasm also. So that AUG will code only for methionine, whether it is in prokaryote or in eukaryote or in any cell. Then non-ambiguous, the genetic code cannot specify more than one amino acid. One codon cannot specify more than one amino acid. Only one amino acid, it can code only one amino acid. Then Non-overlapping. Non-overlapping means, suppose this is a chain in which we have A, U, G. Then we have A, U, G. Now, when AUG will code for one amino acid, it is not that G will code for the next amino acid. This is wrong. Okay, it is always that AUG will code for one amino acid and then the next three will code for the next amino acid. So that is, it is non-overlapping. The gene is transcribed and translated continuously from a fixed starting point to a fixed stopping point. It is continuous. Then the code has polarity. It has definite direction for reading of message, which is referred to as polarity. So reading of message from left to right and right to left will specify for different amino acids. For example, UUG stands for leucine and from right to left it stands for valine. Then degeneracy of genetic code. That means amino acid can be coded for by more than one codon. Here you can see leucine is coded by 2 codons. Again 3-4 more codons leucine. So one amino acid is coded by more than one. So what is this? How does this happen? This is explained by Wubbel hypothesis. He said that according to Crick postulated the Wubbel hypothesis. He said that the first account for the degeneracy. According to this, the first two bases of the codon pair according to the normal base pairing rule with the last two bases of the anticodon. That means anticodon here is GAG. Now this GAG from here, GAG. Now this first G and this last G and A will code with the first C and U of the codon of the mRNA codes. Okay, and the third one is a Wubbel. It can change. G and C, which are changing, this can change. So that is explained by more than one. That explains more than one codon for particular amino acid. Wubbel hypothesis explains the degeneracy of the genetic code, that is existence of multiple codons for a single amino acid. Although there are 61 codons for the amino acids, the number of tRNA is far less, which is due to wobbling. Then biological significance of degeneracy of genetic code. 20 codons will be designated amino acids and 44 would lead to chain termination. The probability of mutating to the chain termination would therefore be much higher with a non-degenerate code. Now, flow of genetic information, we know that DNA transcribes RNA, RNA translates into protein. That means that code, here you can see how DNA will change into RNA, RNA will change into protein. Then clinical significance is that mutation can be well explained by genetic code. We will read about this. Point mutation, silent mutation, missense mutation, nonsense and frame shift. What happens? Silent mutation. Here single nucleotide change A to G. You can see here that this is the A nucleotide which is changed to G. So here what happens? The same glutamine is synthesized. Same amino acid is synthesized. So mutation goes unnoticed. Then that is called silent mutation. Then missense mutation, in which single nucleotide change A to C and different amino acid is produced. So loss of functional capacity of the protein occurs. Now here the A is changed to C. Hence instead of this histamine protein, another protein is produced. Okay. So PRO protein is produced, whatever it is. So what happens? The function of that amino acid is produced. So function of that protein is changed. Now nonsense mutations. Single amino acid, the single nucleotide change from C to T stop codon is generated. So what happens here? When the C is changed to T, it generates a stop codon, TAG. And then what happens? The termination occurs and it is incompatible for the life. Then frame shift mutations, in which one G is removed. Then what happens? New amino acids are produced. First is methionine. Then instead of glutamine, because AAG will combine, CAC will combine. So AAG will give lysine and CAC will give histamine. So this is what was total about our genetic code. Thank you.